In addition to enzymes, some chemical agents, they can also influence the supercoiling of the DNA depending upon its concentration that is used. So one of the chemical agent, it is ethidium bromide. So if we want to analyze the DNA and want to see it under the camera or under the UV light, then after running the DNA on agarose gel, then the gel can be stained with a chemical like the ethidium bromide. So when it is exposed under UV light, the band of DNA that are separated, they can be visualized as orange color. So actually this ethidium bromide, it can intercalate between the base pairs of the DNA. So intercalation means that the reversible addition or insertion of chemical into certain compounds like the DNA. So when it will intercalate into the DNA, then it can also affect the supercoiling of the DNA. For example, if we isolate supercoiled DNA and treat it with ethidium bromide, then after intercalation, the supercoiled DNA, it will untwist. That is, its unwinding will start. So if intercalation it is going on, that is addition of ethidium bromide it is going on, then the supercoiled DNA, it is converted into open circle. That is covalently closed circle DNA or CCC DNA, or simply we can say that it is open circle DNA. But if the intercalation of ethidium bromide it is not stopped at the stage and still it is going on, then the again rewinding of the open circle DNA it will start, but now in opposite direction. That is negative supercoiling it is introduced. And the open circle DNA and the supercoiled DNA and the linear DNA. Linear DNA means that the plasmid it is now digested with single enzyme, so that now it is no more open circle, so it is now in linear form. So if we analyze it on the gel, then we will find that because of its structural configurations, they will separate at different locations on the gel. यानी कि जब हम एथिडियम ब्रोमाइड से ट्रीट करते हैं तो ओपन सर्कल डीएनए या सुपर कॉइल डीएनए की जो कॉन्फ़िगरेशन है, जिसको हम स्ट्रक्चरल कॉन्फ़िगरेशन कहते हैं, वो चेंज हो जाएगी. तो सुपर कॉइल डीएनए और ओपन सर्कल डीएनए जो है जेल पे डिफरेंट लोकेशंस पे सेपरेट होंगे. और इसको हम कंपेयर कर सकते हैं लीनियर डीएनए से भी, कि अगर हम सपोज प्लाज्मिड को एक एंजाइम लाइक EcoR1 से ट्रीट करें जो कि प्लाज्मिड को एक ही दफ़ा कट करे, तो लीनियर डीएनए भी डिफरेंट लोकेशन पे सेपरेट होगा. सो वी कैन से दैट ethidium bromide intercalation it can change the structural configuration of the plasmid DNA. So it is evident from this diagram. So it indicated the electrophoresis of DNA on agarose gel, and comparison is between the open circle DNA and the supercoiled DNA, which we can call it OC or SC DNA. So both type of DNA, they are loaded on a single slot or the well, that's A, and when it will move on the gel, then they will be separated on different locations depending upon the structural configuration. Because of supercoiling, the supercoiled DNA it will move faster, because the pore of the size it will facilitate the supercoiled DNA to move faster as compared to open circle that will move slowly because of its configuration. And if we will load the linear DNA that is represented here as L, that is treated with, suppose, an enzyme like EcoR1, so it will move a little bit ahead of open circle DNA. So depending upon the configuration of the plasmid DNA, so they will be separated at different location on the gel. So this diagram, it indicates the intercalation of the ethidium bromide, and the DNA fragments, they are visible in the form of orange bands, and this is the ladder that is being run on the gel. So visualization of gel under UV light that is stained with ethidium bromide. This is another diagram that showed the effect of ethidium bromide on supercoiling of DNA. So here in figure A, the DNA, it is present in highly supercoiled form, and if it is treated with ethidium bromide here at this step, then it starts its unwinding, so that supercoiling form, it is converted into finally open circle here, which we can call it as the OC DNA. And if the intercalation of ethidium bromide, it keeps on going, then the rewinding of the DNA will start in the step D, but now in opposite direction. Suppose if at step A or B here, if the supercoiling, it is in anti-clockwise phase pose, then in the step D, it may be in clockwise stage. So the intercalation of the ethidium bromide will cause the DNA to rewind again, but in opposite direction, that is negative supercoiling, it is finally there. So this is the effect of ethidium bromide on the supercoiling of the DNA. Although ethidium bromide, it is practically very valuable to visualize the fragment of the DNA after running the gel, but at the same time we must take some precautions because of the side effect of ethidium bromide. Side effects may be that if it is used in higher concentration, it may be mutagenic if living organisms, they are exposed to ethidium bromide. At the same time, it may be a carcinogenic as well, that it can also cause the cancer formation. And depending upon the circumstances, ethidium bromide exposure may be teratogenic, that is having some developmental abnormalities. If some living organisms, depending upon the stage of exposure, they are there. And finally, the use of ultraviolet light, it is also dangerous if someone it is exposed to that radiation, because it can cause the skin or the eyes damage.